Yeah, so this is a great question. This study looks at the comparative safety and efficacy profile of the two FDA-approved CAR T-cell products, idecabtagene vicleucel and ciltacabtagene autoleucel. This is for patients who have multiply relapsed disease, and nearly all of these patients have been triple class exposed to IMiDs, PI, and CD38 monoclonal antibodies. Yeah, so this was a retrospective analysis. This involved about 19 centers in the United States, and this is institutions within the U.S. Multiple Myeloma Immunotherapy Consortium. We essentially included 640 patients who were leukapheresis with the intention to receive a CAR T-cell product. Of those patients, about 55 did not proceed with the infusion, mostly due to disease progression or death. About 586 patients were infused. We had 350 with idecabtagene and about 236 with ciltacabtagene. And in addition to the primary analysis, we performed several sensitivity analyses to confirm the results. And the statistical methodology utilized for the study was inverse probability of treatment weighting. And this is basically a type of propensity score matching to balance measure confounders between comparison arms, and that is we do this type of methodology to preserve cohort size. But really the main reason for this study is, there isn't a prospective randomized controlled clinical trial to look at this. And we have, two single arm studies that have some differences in responses and in safety and in survival. So in lieu of a randomized controlled clinical trial, we did this kind of retrospective analysis utilizing inverse probability of treatment weighting. Yeah, so the main results of the study showed that ciltacabtagene or ciltacabtagene autoleucel was associated with a higher likelihood of experiencing severe cytokine release syndrome, which was grade 3 or higher cytokine release syndrome, a higher likelihood for experiencing infections, as well as delayed neurologic toxicities. There was a trend for increased second primary malignancies, but this did not reach statistical significance. The numbers are small, so obviously this result must be interpreted with caution. We also found that patients receiving ciltacabtagene were more likely to experience a better response, particularly a deeper response. And we also identified that patients receiving ciltacabtagene had significantly superior progression-free and overall survival compared to idecabtagene. And this was overall and within nearly all high-risk patient subgroups. So, as I mentioned, I think these results are important because, we don't have a comparison in clinical trial between these two studies. And because we saw some differences in the single arm studies, I think it's important we identify, what patients are best for which CAR T-cell product, or what patient may benefit better from Abecma versus Carvykti. And we hope that this study, should inform or help with clinical decision making, counsel patients, and hopefully help select or guide selection of CAR T-cell products for patients proceeding with chimeric antigen receptor T-cell therapies for relapsed refractory multiple myeloma.